Welcome to Dr. Glynis' Chicken Genetics videos. Today we're going to look at the gene that controls gold and silver. Ulrika Gunnarsson of Uppsala University in Sweden mapped the silver mutation to near the top of chromosome Z in a membrane-associated transport protein which is known to be involved in colour dilution in other animals. As I understood it, there was a strong suggestion that a separate mutation may also exist nearby, affecting silver in white leghorns and maybe other breeds, so that not all birds have exactly the same mutation. With the mutation being sex-linked, females can only be hemizygous at this gene. They have one allele, which is either the wild-type allele or the pheomelanin-diluting silver allele. Males may be homozygous for either allele or heterozygous with one of each allele. The silver allele is semi-dominant over the wild-type allele and turns red pheomelanin pigment to white when it's homozygous. Hemizygous females are also silver, presumably because they don't have any active wild-type allele to make a protein involved in the synthesis of pheomelanin pigment. Birds that are homozygous or hemizygous for the wild-type allele produce normal wild-type pigment. Silver is an example of incomplete dominance, which is also known as semi-dominance or partial dominance. It is not completely dominant over the wild-type allele because the heterozygote exhibits a phenotype which is intermediate between the homozygous forms. The silver allele inhibits red, but only partially does so when it is heterozygous, leaving a colour like golden straw. This is, strictly speaking, from a genetics point of view, the gold phenotype. As females are hemizygous, the gold phenotype can only occur in males. Now, I'm aware that this could create confusion since in Wyandottes the names are used slightly differently. Gold is the traditional name for the colour of Wyandottes, either homozygous or hemizygous for the wild-type allele, and that's fine and they look lovely. However, from a genetics point of view, gold is the partial dilution of the pheomelanin by the silver allele in heterozygous males. As long as we can remember what's what, it doesn't really matter which convention is used. In a lovely video by a lady who breeds Wyandottes, it indicates clearly that in this breed, heterozygous cockerels are in fact silver, but with golden hackle feathers, and indeed I've seen similar birds with gold also in parts of the wing and the saddle feathers. To my eye, they are strikingly beautiful birds, although I get the impression they are not entirely favoured by Wyandotte experts. I've heard that not all pheomelanin is diluted, and this makes sense of some things we see. The work by Ulrika Gunnarsson, which is very interesting and I'll reference below, suggested that the phenotypic consequences of silver alleles may be highly influenced by the genetic background. We should also remember that her work suggests that more than one allele could be at work, albeit potentially affecting the same protein. We have learned that silver is a partially dominant gene on the sex chromosome Z. We will meet it again in a future video when we consider sex-linked crosses as a means of sexing newly hatched chicks by their down colour. Thank you very much for watching this video. If you have found it useful, please like, comment below and share it with your friends or on social media. Subscribe so that you are notified of chicken genetics videos as they come out. I hope to see you very soon.